Hello everyone. So our topic today is the effect of abiotic stress flooding to the plant cell wall. Our group members is Shazrin, Hafiza, Aini, Amira and me, Rosmida. So let's begin.
How does stress affect the cell wall metabolism for abiotic stress of flooding? Okay, the first is low oxygen level in the root zone due to low diffusion rate of water. What happened? The suppression or reduction of hypothetical and root elongation, suppression of lateral root development. So what happened in detail to the cell wall metabolism? Okay, the first is in polysaccharide metabolism, cell wall degradation and growth are inhibited. The component of polysaccharide metabolism, such as cellulose synthase will decrease. Hemicellulose, beta-glucosidase and beta-galactosidase both will decrease. Pectin, based on FTIR analysis, it showed that the degradation of cell wall polysaccharides and a decrease in pectin content. We take one plant, which is Azuki bean seedling. The cell wall sugar content will decrease and cell wall thinning occur. For lignification metabolism in poplar, the expression of genes on encoding cell wall degradation enzymes such as PAL, transinumate 4 hydrolase, C4H, 4-comerate CoA ligase, 4CL, and the rest are the enzymes involved in lignin biosynthesis. Okay, so in flooding stress, these enzymes are inhibited or repressed. Okay. Proteomic analysis showed because the data concerning flooding stress are limited and the data available in the literature are more related to transcriptome and proteome analysis than to the detailed chemical composition. For the soybean seedling, we can see here the polygalacturonase inhibitor 2-like protein and EXPLP1 protein levels were greatly increased. Cellulose synthase interactive protein 1 and CAD decreased. Inhibition of cell elongation of soybean seedling under flooding and the relative increase in the abundance of EXPLP1.
Next is the effect of flooding to the cell wall component, which is cellulose, hemicellulose, pectin and lignin. First is cellulose. In grape poplar plant, the expression of genes encoding cellulose synthesis required for secondary cell wall formation was strongly downregulated in roots, while those involved in primary cell wall biosynthesis were drastically affected. Second is hemicellulose. It will decrease the beta-galactosidase, decrease beta-glucosidase, and in the flood zone, the C endosperm of Hematitis cuba is richer in mannan. The presence of pure mannan slow down water uptake in seed adapted to the environment. Lastly is pectin. It will decrease the polygalacturonase, increase the polygalacturonase inhibitor protein, less abundant cell wall homogalacturonase in altered pattern, degradation of cell wall polysaccharides, and decrease in pectin content in maize seedlings.
Next, we move on to the changes in the cell wall as means of protection to decrease. Fluoroglucinol staining can be observed suppressed in roots and hypocotyl of soybean seedlings. Next, gene expression poplar encoding cell wall degradation enzyme is repressed. Gene encoding lignin biosynthesis enzyme PAL, transinamate 4-hydroxylase C4H, 4-coumarate CoA ligase 4CL, ferulate 5-hydroxylase F5H, COMP and CoAOMP is indicate the alteration of the lignification biosynthesis. Next, downregulation of an extracellular superoxide dimutase CUZN. It's play a like protein with oxalate oxidase or superoxide dismutase. Activity produce H2O2. It is used in cross-linking reaction within the cell wall. It indicates a reduction in the linking of polysaccharides and protein within the cell wall and liquefaction. In addition, under cladic stress, there are a few substances decrease in lignin. First, decrease in phenylalanine ammonialyase (PAL), which is the important plant enzyme that eliminates ammonia from phenylalanine to form tranexamic acid, a precursor of lignin flavonoids and coumarins. Next, decrease in hydroxycinnamolyl alcohol dehydrogenase (CAD), which is an enzyme specifically involved in the last step of monolignol biosynthesis. It reduces the hydroxycinnamaldehyde to their corresponding hydroxycinnamyl alcohol before transport to the cell to the wall for liquefaction. Decrease the tranexamic 4-hydrolase C4H. Decrease the 4-coumarin CoA ligase 4Cl. Decrease the ferrylate 5-hydroxylase F5H and decrease the caffeine-O-methyltransferase COMT and coffeol-CoA 3-O-methyltransferase CoAOMT. Monolignols, which are the building blocks of lignin, are synthesized from phenylalanine through the general phenylpropanoid and monolignol specific pathway. It involves cytosolic enzymes including phenylalanine ammonialyase (PAL), 4-coumarin-CoA ligase, cinnamyl-CoA reductase, caffeine-O-methyltransferase, and caffeol-CoA 3-O-methyltransferase, as well as ER membrane-anchored protein including tranexaminate 4-hydroxylase C4H and ferrylate 5-hydroxylase F5H. The monolignols are then transported to the wall where they are polymerized by apoplastic peroxidase (PRX) and lacases into lignin. Its adaptive mechanism based on the decrease in cell wall expansion and cell extensibility, which can limit water loss and prevent cell collapse due to dehydration. However, in this case, during flooding, the plant reduced the lignin deposition due to waterlogging by decreasing all the substance.
As we know, when flooding, oxygen level will decrease because oxygen content in water is much lower than air. So, what can enhance gas exchange underwater? A research paper, rice leaf hydrophobicity and gas film are conferred by a wet synthesis gene LGF1 and contribute to flood tolerance. In this experiment, they used rice methane dripping wet leaf 7, which does not retain gas film on leaf, and its wild-type king meat. In gene discovery for this tree, gene complementation was tested in transgenic life, and underwater photosynthesis were evaluated. Rice leaf is hydrophobic, but when submerged, the gas layer diminishes. Leaf gas film 1 is a wet synthesis gene that can retain the leaf gas film on leaf surface. LGF1 regulates C30 primary alcohol synthesis, which is necessary for abundant epicuticular wax platelets, leaf hydrophobicity, and gas film retained on leaf. Hydrophobic body surface is a mechanism to enhance gas exchange with water. So, LGF1 is essential for survival of rice and can improve submergence tolerance. This gene can introduce the plant in flood-prone areas.